there are work on both directions.
Okay, the time was sent. And again, thanks for your intervention. That was really amazing. And we really appreciate it. So now let's give a warm welcome to Dr. Sanjairaj, who works at the New York University in Abu Dhabi. He also contributes his expertise as principal investigator on the BHI lab, giving more than 60 publications to literature about bioprinting. Today, he will enlighten us on a super interesting talk about challenges and future of bioprinting.
Thank you for the warm welcome. I hope you can hear me well. You can see me, my presentation.
Yes, we can see your presentation. I hear you well.
Wonderful. Thank you so much for the introduction. And I think it's a very good continuation from the previous speaker, transitioning from biomaterials and challenges into the bioprinting. So just before I start the presentation, just a brief introduction to the university that I'm from. So I work for New York University, Abu Dhabi. So we have 3 degree granting campuses of New York University, of course, one in New York, one in Abu Dhabi, where I'm located at, and one at Shanghai, China. And we also have 11 other academic centers, right from Accra, Ghana, Argentina, to Tel Aviv, Israel, and so on. This is our beautiful campus here at Abu Dhabi. So if you guys are passing by Abu Dhabi, you're welcome to the lab. Just to give a snapshot of our students, we have 2,100 plus undergrad students, 115 plus graduate students, and NYU Abu Dhabi is one of the most diverse campuses in the world, where we have students from more than 125 plus countries on campus. So it's kind of a microcosm of the world, and I love the diversity here. With that, I'll just move into my lab and what I do here and we as a team do. So my lab focuses on 3D printing and bioprinting for tissue engineering, regenerative medicine, drug testing, and medical devices. And what you see here on this slide are a few of the projects that we have done in the past. So the one you see here on the top, these are nerve guide conduits for peripheral neural injury for treating them. And then we have worked on skin bioprinting. This is a meniscal scaffold for tendon repair and so on. And this pink structures that you see here, these are bioprinted human skin for cosmetic testing. And this project was sponsored by Procter & Gamble Singapore. When I was with National University of Singapore, we worked on that project. And then these structures that you see here are ceramic structures, and these are called triply periodic minimal surface structures. And we have done quite a few works on how to use these kind of structures to create biomimetic bone scaffolds and implants. So with that brief introduction of my lab, just a brief introduction to the field of additive manufacturing and the medical applications of it, so that you understand when I go into the challenges that I talked about later, so you get the context first. So if you want to group the medical applications of additive manufacturing, we can do it in 4 buckets. So the first group of applications would be educational models, where you can create these 3D printed anatomical models for student education and patient education. And the field of 3D printing had grown so much that you can print these anatomical structures with organoleptic properties. So what do I mean by organoleptic properties? You can print these models with the same property as that of the native tissue, right? How this is useful? Probably, the students or the patients can understand how their anatomy looks like, the anatomy of a particular organ. And then they can even like feel and touch and say, for example, for the next set of applications, if they want to do a pre-operative planning, so that surgeons can even cut and see like how much pressure they have to apply to make an incision in the heart, for example, right? So that is an advantage of 3D printing anatomical models. The second group of applications are surgical models. And like I said, for complicated surgeries like pediatric cardiac surgery, where it takes, sometimes close to 24 hours of continuous surgery, it's very difficult to take split-second decisions after opening up the heart because, it's not an adult heart. We are talking about pediatric patients here. So if you can, from the MRI or CT scans, print out anatomical model, you can do pre-operative planning. You can think about, okay, this is where I'm going to make the cut. This is how I'm going to put the implant, or this is how I'm going to do the procedure, and this is how I'm going to stitch it up, right? So you can do pre-operative planning, surgical simulation, and you can also 3D print customized surgical tools and guides, because depending on the patients and the condition that they are in, you might have to have specific surgical guides. Similarly, depending on the surgeons, you might have to have specific customized surgical tools which are easy for them to handle during the surgery. Third group of applications, medical devices. I don't have to talk much about it. All of us know about wearable devices, prosthetics, and implants. And the last and the most exciting part of medical applications of additive manufacturing would be tissue engineering, where it can be scaffold-based or it could be bioprinted tissues, right? I'm going to talk about it in a bit. And if you look at why do we need a three-dimensional structure, there is always a difference between two-dimensional cell cultures and three-dimensional cell cultures. So to give you an example, there are many drugs that are being tested, right? Say, for example, cancer drugs. Many drugs would actually kill the cancer cells when they are actually cultured on 2D substrates. The efficacy is really great. But then the moment you transfer from 2D to 3D in terms of animal models, the drug which performed very well in 2D in vitro cultures would fail miserably when it is inside the animal, right? For the simple reason that the cells are not in the same environment. Cells in 2D are not representative of cells within the human body, right? Because humans, as we are three-dimensional beings. So we need a three-dimensional microenvironment for the cells to feel at home, right? So that's the main reason why 3D printing or 3D bioprinting is required. Second, there is a growing organ shortage. You can see the numbers here. This is just in the US, but we have hundreds of thousands of patients waiting for organ transplantation every year, and the numbers are only growing, right? So if we can, with all our expertise, can create or fabricate organs in the lab, that is a huge achievement, and it would be massive in terms of saving patients' lives. And another reason is not just for the organ transplantation stuff, but also for other testing, right? We do cosmetic testing, we do drug testing. For all that, we are using animal models here. And there are many ethical concerns surrounding animal testing. So bioprinted tissues are viable alternates, right? So with that introduction, I would go into the two approaches that I talked about earlier. One is a top-down approach or scaffold-based approach, where you can 3D print a scaffold. I use 3D print here because there are many other methods by which you can create these three-dimensional scaffolds, but because my talk is focused on 3D printing and bioprinting, I would rather use that. So you can have a 3D printed scaffold, which is acellular, without cells, only biomaterials. And then you can introduce cells into the porous scaffold structure, and then you let the cells grow and proliferate, and they deposit their extracellular matrix, and then it grows into a tissue construct. You can keep it in a bioreactor and so on. So that's one approach, which is scaffold-based approach. The other approach is bottom-up approach, where you kind of use cells as building blocks to create tissues, right? So either you can do cell aggregates, you can do cell sheets and stack them, or you can do cell laden hydrogels, or you can even do cell printing, right? So this is another way of building tissues as opposed to the scaffold-based method. So with that, an introduction to bioprinting. I think all of you guys by now know what is bioprinting. So bioprinting is the use of 3D printing technology that incorporate viable living cells, right? So this is very important. Anything without cells, we don't call it bioprinting. It is just 3D printing. You may ask, oh, I'm just printing biomaterial. So can it be called bioprinting? Actually, no. So it could be called biomaterial 3D printing, but it's not bioprinting. So the moment we use the term bioprinting, living cells are part of the bio ink, as we call it. And as you can see from the process here, there are 3 stages of bioprinting, and some are relevant, some are not relevant, depending on the application that you're working on. So usually there is a pre-processing step where you do all your imaging like MRI, CT, ultrasound, etc. And then from those images, if you want, you can get a 3D model and prepare the STL file for 3D printing. So now there are new file formats like AMF and stuff, but I won't go into that now. So that those are the pre-processing steps. And then you have the processing step where you kind of take the harvested patient cells or any cell lines, commercially available cells, depending on your application. You do the culture, you kind of create the bio ink, and then you do the actual bioprinting process. So that's the processing step. And then you have a post-processing step where either for static cultures, you just keep it inside the incubator, or you can use a bioreactor for tissue maturation, where it's more dynamic and mimicking the physiological conditions. And then when it comes to application, like I told before, it could be in vitro models for disease modeling or drug or cosmetic testing, or it could be for regenerative medicine purposes where these constructs are being transplanted into human beings or patients or animals, right? So again, I won't go into all the details of it, but there are many types of 3D bioprinting. So it could be laser-based bioprinting, where laser is used to pattern cells. You have droplet-based bioprinting, where it could be inkjet bioprinting, or it could be electrohydrodynamic jetting, acoustic bioprinting, microvalve-based bioprinting. So all these are droplet-based bioprinting processes. Then you have the most commonly used bioprinting method, which is extrusion-based bioprinting. And then you have stereolithography bioprinting, where you use photocross-linkable gels and you use light, some kind of a light source. It could be UV, it could be visible light, it could be any other form of light that's used to photocross-link the gels and form 3D structures. And then one emerging bioprinting field is the volumetric bioprinting, where you even have a couple of commercial bioprinters available now, predominantly from Europe, where the constructs or tissues are built up from all 3 dimensions in a volume fashion than layer by layer methods as opposed to the other bioprinting methods. So which makes it faster and more efficient. So again, just to clarify certain terminology before I go into some of my research, and then I touch upon the challenges of bioprinting. You have to understand the difference between biomaterial inks and bio inks, right? This is very related to what I talked about earlier, the difference between 3D printing and bioprinting. When you talk about bio inks, cells are a mandatory component of the printing formulation, right? Without this, yes, it is a biomaterial, but without cells, these can be called biomaterial inks, but not bio inks, right? So that's important to understand here. Then I'll talk about a few of the projects we have done in the lab recently, so you get to understand what kind of projects that we work on. And then I'll go into the broader field of challenges and the future of bioprinting, which I think is very important for now to understand at this point of time on Earth. So we do work on eco-friendly bioinks from various sources in our lab. So I'm going to talk about like 2 such projects very briefly without taking a lot of your time, just to give you an introduction. So the first project that I would like to talk, or a series of projects, is based on what are called tunicate-derived decellularized extracellular matrix bioinks. So I'll refer it to DECM bioinks from now. For those of you who have no idea what these organisms are about, so these are called sea squirts or ascidians at different parts of the world. And the problem with them are these are called invaders or fouling tunicates. So they are non-native species. They travel from one port to the other. And because they are filter feeders, they kind of take over the local marine ecosystem. And because of their high temperature and salinity tolerance, they kind of eat all the food, and the local species are kind of disadvantaged, and they kind of disappear from that coast. So these are, in a way, a threat to marine ecology. Not all species, but some of the species of these, right? So what we did is to take these organisms, and then we kind of explored if we can use them as DECM scaffolds or bioinks for bioprinting. So what we did is we took these organisms, and then we took the outer skin of this organism. These are called tunics. And then we kind of lyophilized them and used them as a DECM material. Now, we tried three different routes, three different applications for doing it. The first one we tried was to use them as a wound dressing material, right? So we did a lot of experiments with that. And the cool thing with this material is the capacity of this material of absorbing 20 times its weight in the dry state, which is 2 times that of the commercial wound dressing materials, was amazing. So we could use them as wound dressing material. So that's the first application we tried. The second application was, can we use them as a natural DECM scaffold? Can we culture cells on them and see how that works? And it worked amazingly. And one important advantage of these DECM material, I would say, is the mechanical property. If you look at the normal hydrogels in the literature, they range anywhere between 0.1 to 1 MPa. I'm talking about tensile modulus. And even to reach 1 megapascal, you have to do a lot of nanomaterial addition into it, nano clay and stuff like that to make it stronger. Otherwise, hydrogels are weak materials. But this DECM material, though it looks very much like a soft tissue hydrogel material, it has an impressive tensile modulus of 4 megapascals. And then the third route that we tried is to formulate a bioink out of it. And to reiterate again, bioink contained cells. And in this case, we used mouse embryonic fibroblasts. And then we were able to print using extrusion bioprinting. And we studied all the viability and other biological characteristics of the cells. And we found, it could be used as a biomaterial for bioprinting as well. And we also explored can we use this particular DECM material for more specific kind of tissues. So the first thing we tried was for cartilage tissue engineering. And we kind of compared between a DECM scaffold and the bioprinting of these inks with human mesenchymal stem cells. Right. So as you can see from the images here, first we cultured them on DECM scaffolds. Everything was fine. And after we culture, we also differentiated the human mesenchymal stem cells into chondrocytes. And we have done extensive studies, including staining and immunostaining and PCR studies for gene expressions. And we were able to successfully differentiate the HMSCs into chondrocytes. Right. So these are all the stainings that I talked about. These are the different gene expressions that I talked about, which confirm that we have differentiated these HMSCs into chondrocytes. And this was done both on DECM scaffolds and on bioprinted tissues. And then a step further, we thought, can we use them for neural tissue engineering? So there is something interesting here, which I want to mention. When we used the DECM scaffolds and when we cultured the human neural stem cells on these scaffolds, everything was fine. So as you can see, the cells attached well, the cells grew well, proliferated well. and we initiated the differentiation, and we were able to differentiate the neural stem cells into peripheral neurons, right? So as you can see from the gene expression studies here, HNK1 is a stemness marker, so it reduced. Beta-3 tubulin is pan-neuronal marker, it was upregulated. Peripherin and neurofilament heavy chain are peripheral neuron-related markers, which are also upregulated. Everything was fine. We did not use any kind of coating. And for those of you who are in the field, to culture these stem cells, we usually coat the surface with a matrigel or some kind of a basement membrane proteins so that the cells will adhere and attach and proliferate and grow. So on DECM scaffolds, we did not use any such coating and everything was fine. But the moment we used the bio ink, the moment we took the route of 3D bioprinting, extrusion 3D bioprinting, as you can see from the images here, D, E, and F, the cells died. The cells did not live. And remember, this was the exact bio ink composition that we used earlier for printing human mesenchymal stem cells. So what worked for MSCs did not work for NSCs. So we had to add matrigel as a component of the bio ink. So in fact, it was a matrigel bio ink rather than a DECM bio ink at the end, because the concentration of matrigel we used was 26%. So that was the optimized value, both for printability and also the cell, the biocompatibility and the cell growth and proliferation. So it ended up having 26% matrigel and 10% of the DECM material. And yes, we were able to successfully print them, as you can see. And we also successfully differentiated them into peripheral neurons later. So the point here to understand is, the bio inks, though there are attempts to do universal bio inks, which I'll talk about when I talk about challenges, we have to tune the bio ink based on the type of tissues that we are targeting, the type of cells and tissues that we are working with, right? And yeah, I just want to mention about this slide. So when we talk about bioprinting, we also have to consider, can we store and use these tissues or organs? Because, it's not a cake to bake and immediately use it. It doesn't work like that, at least till now, for bioprinting or biofabrication. So the time is very crucial. The preparation time and the printing time and then the maturation time, everything is very crucial, and it cannot be done in days, right? It's usually, a few days to few weeks or even few months, depending on the scale of the tissue, type of the tissue or organ that you are working with. So what we did is, we took these DECM scaffolds on which we seeded and differentiated the NSCs and also the bioprinted neural tissues. And we kind of saw, can we store them and again, like rethaw them and see if the cells would survive? That's exactly what you see on this slide. Yes, the cells lived, the cells were able to revive, and the structure was still okay. And, this is kind of a proof of concept study to say, yes, probably the bioprinted tissues can be stored and used for long-term storage and clinical transportation, right? So that's a very crude way of doing it, but this is just a beginning, proof of concept study. Much has to be done in the future as well. So that's like one line of a few projects that we worked on. The other one I would like to highlight very briefly is how we isolated collagen from fish skin and cellulose and nanocellulose from the banana stem that you see. And then we tried to use them to 3D print and bioprint a few constructs. So in this case, we took up the model of what are called the tunneling wounds, right? Usually the skin wounds are very superficial, and the available solutions in the market are for treating superficial wounds. The moment I talk about tunneling wounds, we are talking about wounds that are deep and then not really vertical, but it could even go horizontal. So that's why it's called tunneling wounds. It could be like tunnels underneath the skin, which are most difficult to treat. And if you do not treat it, it could pose major health concerns. So that's where we worked on fabricating what we call tunnel wound fillers with the fish skin that we isolated, with the collagen that we isolated from fish skin and the cellulose that we isolated from the banana stem. So both, if you look at it, they are like waste products. So literally these banana stems came from my kitchen. So these are my kitchen waste. And if you know the Indian style of cooking, we use the banana stem for cooking some very nice vegetarian dishes, right? So literally these are my kitchen waste. And these fish skin, we collected it from the port where they were, cleaning the skin and everything before selling the fishes. So both are tailored towards sustainability and circular economy. So without going much into the details of it, it's already accepted. So soon it will be published and you'll be able to read it. But the idea here is we kind of used the cellulose microfibers and then we kind of coated it with different layers of collagen, proving that, we could fabricate drug-loaded tunneling wound fillers, which could be used to treat these very complicated tunneling wounds. So here you see those bioprinted plugs. These are with cells. If you would want with cells, you can do it. And this one you see here is with a drug, like baniosin is a common drug used for wound healing and stuff. So you can see how the drug can be released over time using the different coatings of the collagen on the cellulose microfibers, right? So again, this is just a proof of concept to prove the structural integrity and ease of handling these tunnel wound fillers that we created to keep it inside and take it. So we just used a chicken wound model just to see if we can handle these tunneling wound fillers to treat and to dress and like remove it and replace it and so on. So with that brief introduction to some of the projects in my lab and a general field of bioprinting, I would like to conclude probably in the next 5-10 minutes maximum some of the challenges that are concerning the field of bioprinting, which if overcome would be the future of bioprinting. So let's start with bioprinters. Now, there are many bioprinters in the market now. So we are talking about scalability, multi-axis movement, multiple printing modalities, etc. So all these, I'm quite sure the companies will take care of it. And we as researchers, many are working on it. So that I don't have any problem. But one challenge that's a real challenge is the scalability, because maintaining the cells without damage during this long printing process, it is really a challenge because bioprinting is a slow process, right? All of us know it. If some papers, some speakers tell you, oh, bioprinting is very efficient, no, it is still a slow process. So with scalability comes the challenge of cells survived during the long printing process. Right? Imagine you have to print a heart, right, of this size, and it takes, say, 3 or 4 hours to print that heart. We are talking about cells being printed, right? So how are we going to maintain the cells without damage during this long printing process? So of course, volumetric bioprinting or fresh bioprinting, if you have heard of those bioprinting methods, we are trying to address it by making the bioprinting process faster, right? In terms of fresh bioprinting, you are actually printing it within a bath. So probably you can use cell culture media and stuff to kind of preserve the cells from damage. So that's some of the techniques that could work. And another associated challenge will be in situ monitoring, right? As we are printing, can we quantify the health of the cells in some way? Because during this long process, what if the cells start dying at the bottom, right? As you are like building the tissue or organ up. So you can at least, make some course correction if that is the case, if you have systems for in situ monitoring. The other one is the clinical friendly bioprinters, right? I mean, we like complex bioprinters with a lot of options. But is it really necessary? Is it really helpful if you have to make it available to the clinicians? So that's a question that we must ask. Now, coming to bioinks, the first and the most important challenge is the source of bioinks, right? If you look at the market today, most of the hydrogels or bioinks, their source are animal derived, be it collagen, be it gelatin, be it fibrin, right? I mean, there are other plant derived hydrogels as well, like alginate or agarose, but predominantly there are animal sources of collagen. And animal sources of bioinks and hydrogels. So that might cause potential harmful immunological reactions. You might say, oh, we did a lot of experiments in the lab. The cell seems to like it, love it. They are, happy being in those hydrogels. But, I'm giving an example with a reference here. The ECM product core matrix, which although approved for use by several regulatory bodies in different jurisdictions, showed significant chronic inflammation and fibrosis in the long term. So, we really have to do a lot more studies to understand the immunological reactions and effects of these animal-derived sources of bioinks. So we may have to do some kind of studies for quantifying the endotoxins or other immunotoxic compounds and methods to decrease their levels if this is the only way to go forward. The other issue with bioinks is the batch-to-batch variation, right? Almost all the commercially available natural polymers have this issue of batch-to-batch variation, right? Because each animal is different, so the ECM materials taken from the animals will be different. So one example is the age of the animal, right? Depending on the age of the animal, the quality of the ECM that you are taking out is going to be very different. So, and then environmental factors, plus the process-induced variations. How does it affect the chemical composition or structure, molecular weight, mechanical, rheological properties, degree of substitution or functionalization, etc. Right? So these might also contribute to the batch-to-batch variation. So that's the second challenge to deal with the bioinks. The third one, which I already talked about, is tissue-specific bioinks. Now, there are efforts to develop universal bioink, which kind of suits all kinds of cells, all kinds of tissues. But I think nothing solid has materialized yet commercially, and still you have to do tissue-specific bioinks. Other challenges, most of the commercial bioinks pass through only basic cell viability tests. So, they just do live death staining for 7 days. They do MTTSA, MTSSA for 7 days. And then they say, oh, this is a super bioink. But, there are many other cellular functions that might be influenced by the bioink, right? So the bioink, I've given an example here. The bioink might support post-printing cell viability, but how about differentiation if you want to try some stem cells in it? So that's an important question to ask. That's something important to study. And then concerning the long-term studies, right? Not just 1 day, 2 day, and 7 day studies, but can it go on for months and see how the properties of the bioink will change with time, be it degradability, be it breakdown of crosslinks resulting in structural failures, or the interaction of new ECM produced by the cells with the bioinks and what kind of effects these things cause inside the body. And the other one, studies in large animal models are necessary because you look at all the bioprinted tissues or organoids that are being tested today, mostly they are done with small animals, mice, rat, probably rabbits and stuff, but not so much in large animal models. So if you're trying to translate this to human scale, of course, we need to look at large animal models. And then the other thing is bioprinting parameters and standardization. If you're working in the field, you would know what I'm talking about. If you even follow exactly the same protocol, exactly the same bioink, sometimes you cannot replicate what has been stated in a particular paper. And even within your lab, depending on the time of the year, the printing might be very different. We face that. I mean, during winter, The printing is very different. During the summer, the printing is very different. So the temperature, the humidity, and anything, any parameter that we normally do not, carefully tend to or kind of report in the literature, that might play a huge role when it comes to printing of these tissues, right? So I suggest, if we can have an open database of bioink optimization, that will really help the field progress to a greater extent, right? And then coming to the cells part of it, I only have like 4 or 5 slides left, so I'll finish it quickly. So the source of cells again. If the cells are patient-derived, that's the best case scenario, right? But then if you look at the time that I talked about earlier, it cannot be used for emergency situations. A person who really needs some kind of a tissue structure to immediately operate upon. We cannot wait to take the cells, grow them in the lab, get 1 billion cells, and then kind of do a bioprinting. Probably the patient will not live to that time. So what is the other alternate? Yes, induced pluripotent stem cells could be a very promising alternate for patient-derived cells. So for those of you who do not know what are induced pluripotent stem cells, so these are stem cells that are created from well-differentiated cells. For example, your skin cells or fibroblasts, you can take them and induce them to become a stem cell, and then that stem cell could be used to differentiate them into any kind of tissues. So it could be bone, it could be muscle, it could be a nerve, etc. Right? And even with induced pluripotent stem cell, one way is for each patient, you kind of have a iPSC bank. And in case of any emergency, you can just use them and culture them and kind of print the tissues. But even better is if we can develop an immune neutral stem cell type, or synthetic or genetically modified iPSCs. If we can achieve that, then you can have what are called the off-the-shelf bioprinted tissues and organs that could directly be used in case of emergencies, right? I mean, all I'm talking about is well further, probably decades further from now. But yes, we are scientists and researchers, so we can imagine and dream into the future, right? Other challenges, associated technologies. For example, bioreactor for tissue maturation. So we have so much development in terms of bioprinting, but are we focusing on bioreactor, which is kind of important if you have to do a dynamic culture of these tissues or organs that you are printing? And at industrial scale, if you are talking about making bioprinted organs available for transplantations. And then there are many other challenges that I could talk. Like one is having a serum-free media, right? So this field of cultivated meat that has like brought in a lot of energy into serum-free media and some of the challenges in traditional tissue engineering and bioprinting that we are talking about, right? And then last but not least, all the technical side of things are good, but we have to think about the ethical, social, and legal policies, because you can do so much in the lab, but if the social perspectives are very different, and I give some of the questions to ponder, when we talk about biofabricating or fabricating an organ in the lab, questions like, can humans play God, naturally comes. Moral deterioration due to technology. So now that I can get a new pair of lungs, will people start smoking more? Now that I can get a new pair of liver, will people start like drinking too much? So these are some questions with respect to moral deterioration, ownership of the printed organ, restricted versus open use, and the social aspects, the religious, cultural views, and social acceptance. These I think has to be considered in tandem with the technological developments if you have to like really translate bioprinting and bioprinted organs into the clinics and available commercially. So with that, I would like to thank and acknowledge all the team members, both past and present, who did all the hard work. And I would also thank the funding agencies and my sponsors. And with that, thank you so much, and I'll be happy to answer any questions you have.
Okay, doctor, thank you very much for this incredible talk. I think you make possible understand this wonderful new knowledge area, and we think it's incredible new area who makes possible things that before we cannot even imagine. So now, let's get time for any questions. Remember, you can make it by text or turning on your microphone.
Yes, I know that there's a kind of thing that we can do with plants. For example, we can cut a stem on a plant and transplant it to another plant. And I guess that, well, thought that it could be possible to use that kind of technique for growing new tissue. In case of animal tissue, for example, we can make an special container where we can use another tissue which is being already germinated, and we can use that tissue to grow another tissue because it is going to get some nutrients for that lower tissue. And it's kind of an idea that I got because of that stem transplantation from plants. I don't know if that technique could be made a similar way to help by that hurdle you said that we had with the time that we spent on growing new tissues.
That's a wonderful question, David. So, I mean, people have tried it already. So they call it in vitro bioreactor. So you can search about it. There are a few papers about it. So what they do essentially is, use a specific region within the body, even like animal body, where, they have specific conditions that could help form a new tissue. And then, they kind of take this tissue out and can be transplanted where it is required. So they have tried that, but, it's not as simple as, grafting that we do with the plants, because here we are dealing with a lot of different types of cells and a lot of different types of growth factors. I mean, I'll give one example. Just this example of matrix stiffness. So for example, you have a surface and then you have stem cells, right? So this surface, if it is harder, the stem cell will differentiate into a bone. If this surface is like very soft, this would differentiate into a nerve lineage, neural lineage. And if there's somewhere in between, it could grow to be a cartilage, or it could be a muscle, etc. So this is just like one factor, which is surface stiffness. And similarly, there are like many other factors that determine how the cells are going to behave, differentiate, grow, mature, etc. So yes, I think the idea itself is like very interesting. People have tried it before, but when we are talking about full-scale organs and scalable tissues, probably, yeah, I mean, this might not work when we talk about industrial scale of fabricating tissues and organs. But I think at a lab scale for experimenting and testing, I think this is a great idea, and that's where they came up with concept of in vitro bioreactors. Thanks, David, for the question.
Thank you for your response.
Other questions?
Anyone have other questions for Doctor? Well, I think that's no more questions for public. So thanks again for your presentation. That was very amazing, and you can make possible to everyone to understand this wonderful topic. So now we're gonna share with you the knowledge quizzes, which are brought up all talks made in this event. Remember, we have a secret award for the better result from Panamericana. You can make this and have 50 minutes to answer it. After it, we're gonna review the answers, and in 1 hour before the end of the questionnaire, we're gonna announce the winner and the prize. So give me a second. I will share to you the question link.
asume todos los costos para realizar conjuntamente con la asociación la publicación de libros, la generación de cursos y programas educativos y otras actividades de formación continua y actualización profesional bajo la modalidad online o impresa. Editorial Médica Panamericana acompañará a la asociación en los eventos que realice, otorgando excelentes beneficios a sus asociados durante el evento. Editorial Médica Panamericana hará difusión en sus redes sociales a sus más de 42.000 contactos del convenio establecido y de los eventos que la asociación organice. La cobertura será a nivel nacional. Los afiliados podrán adquirir nuestros productos a través de la página web con envío gratuito y en nuestras oficinas. Contarán con varios medios de pago y fácil financiación para los productos de formación continua. Juntos llegamos más lejos. Editorial Médica Panamericana. La salud, nuestro proyecto editorial.
¿Vas a empezar a preparar el examen MIR? Te presentamos PROMIR, un nuevo entorno virtual de aprendizaje que te ayudará a obtener los mejores resultados donde y cuando quieras, sin necesidad de desplazamientos. Con PROMIR, el estudio se realiza de forma simultánea al entrenamiento de las habilidades necesarias para superar con éxito el examen. Seguirás un itinerario formativo personalizado, con una metodología estructurada en 4 fases que te ayudará a optimizar tu esfuerzo. Este nuevo y revolucionario sistema de preparación para el MIR cuenta con contenidos enriquecidos y personalizables. Podrás subrayar contenidos, incluir anotaciones y marcadores, e imprimir los materiales y tus apuntes siempre que los necesites. La plataforma contiene más de 30.000 preguntas, que incluyen las de los 12 últimos años de la prueba. Al mismo tiempo que estudias, podrás poner a prueba tus conocimientos con entrenamientos tipo test y simulacros de examen. En tu camino hacia el MIR, contarás con la tutoría y asesoramiento de profesionales expertos en formación médica, que resolverán tus dudas y te orientarán para mejorar tu técnica de examen. Además, contarás con videoclases de apoyo al estudio. La metodología de PROMIR se adapta a ti en función de la convocatoria a la que te quieras presentar y al momento en el que ingreses en la plataforma. Se estructura en 4 fases: calentamiento, construcción, consolidación y competición. Dependiendo de la fase en la que te encuentres, PROMIR te indicará cuáles son tus objetivos de cada día y adaptará los eventos a tus necesidades y a tu ritmo de aprendizaje. El sistema inteligente analizará tu evolución, tu nivel en cada asignatura, el tiempo que has dedicado al estudio y al entrenamiento e irá adaptando los contenidos para maximizar tu esfuerzo y mejorar tus resultados, a modo de entrenador personal. PROMIR es un entorno virtual de aprendizaje totalmente vinculado y multisoporte. Así podrás estudiar donde y cuando quieras, desde cualquier dispositivo. Ponte a prueba, consigue tu acceso gratuito y descubre por fin una nueva forma de preparar el MIR. Infórmate en www.medicapanamericana.com/PROMIR. PROMIR, un proyecto original de Editorial Médica Panamericana.
Give me one more minute. I am fixing something in the questionnaire and I will share it into the chat. So you can scan this QR for register your assistant to this. At this moment, I shared the link of the questionnaire for the chat of this call. You got 50 minutes to answer it, and after it, we're gonna review that and share with you the results in 1 hour after it ends the questionnaire. Thank you.